Hello everyone. In today's lecture, we are going to see about what is artificial blood and how it used, what are the types like that we are going to see. Before we start, let's quickly recall what is blood. Blood is a special connective tissue that performs two main functions. It carries oxygen to all parts of our body and helps defend against infection. And historically, blood transfusion started way back in 1667, but main yearly experiments failed because of fetal reaction. Today, blood transfusion is a routine medical process, but it still faces several problems such as limited availability and infection risks. So scientists started thinking, can we make an artificial form of blood that can safely carry oxygen? That's what we'll explore today. And in today's section, we go through five main parts. First, I will give an introduction to artificial blood. Then we will understand why we need it and understand the empathize stage and next define stage. And then artificial blood, what should do, and after that we look at technological approaches, how it made using materials like perfluorocarbons and hemoglobin based carriers. Finally, we look at testing limitations and current status. By the end of this class, you will understand how biomedical scientists are trying to develop artificial substitute for the real blood. Now let's think, why do we even need artificial blood? Natural blood can only be stored for about 1 month, which makes it difficult to manage during emergencies. There are also safety concerns such as the risk of transmitting disease like HIV and hepatitis through the blood transfusions. Additionally, matching blood groups is always necessary. Otherwise, the blood will reject it. Because of these challenges, scientists wanted to create a universal blood substitute that is safe, long-lasting and compatible with all blood types. This is the main reason behind the developing artificial blood. In this stage, we define what exactly artificial blood should be. It acts as a substitute for the red blood cell, which means its main job is to carry oxygen to the body and remove carbon dioxide. An ideal artificial blood should be safe, free from any infection, and work well with all blood types. It should also be stable for more than a year when stored. I learned that if scientists can achieve this goal, artificial blood will be a life-saving solution in hospitals. And now let's look how artificial blood is being developed. There are two main approaches, perfluorocarbons and hemoglobin-based oxygen carriers. PFCs are man-made chemical components that can dissolve large amounts of oxygen. They don't react with the body and are completely stable. And hemoglobin-based blood, on the other hand, use of hemoglobin, the natural oxygen carrier found in the red blood cell, but without the cell itself. These are modified or purified so they can work safely inside the body. Both approaches try to mimic the function of real blood in different ways. Let's understand PVCs. In simple way, they synthetic fluorinated components that carry oxygen by dissolving it, not by bonding chemically. When used, PVCs are made into tiny droplets called emulsions or injected into the body for them to work effectively. The patient must breathe oxygen-rich air so that the more oxygen can dissolve in the PVC. The advantage is they are non-toxic, don't cause infection, and blood type independent, meaning they can be given to anyone. They also have a long shelf life. Limitation is they carry less oxygen than the real blood, stay in the body only for a few hours, and sometimes cause mild flu-like symptoms. Examples: fluorosol, DA, and oxygen. And then the second major type is hemoglobin-based oxygen carriers. These are made using hemoglobin taken from humans, animals, or produced using genetic engineering. Hemoglobin carrier oxygen naturally, so these products transport oxygen directly through the plasma even without the red blood cells. These are two main production methods: chemical modification, where hemoglobin is cross-linked or polymerized, and the recombinant DNA technology, where it is produced by microorganisms. The advantage is high oxygen carrying efficiency and no blood type required, and can be sterilized and stored long term. The limitation is short lifespan in circulation and causes vasoconstriction and hypertension. Now let's see how the artificial blood is tested and produced. There are three main methods. Synthetic method, where the scientists create oxygen carrying components in the lab, and chemical isolation, where the natural hemoglobin is extracted and purified, and the third technology, where bacteria are genetically modified to produce the hemoglobin. Currently all these methods are still under research and clinical testing. No product yet work as efficiently as real blood, but scientists are continuously improving oxygen delivery and reducing side effects. Even though progress is being made, these are still major challenges. So far, no artificial blood is completely safe or effective for long-term use. The main issues are toxicity, short lifespan, or instability in circulation. Which means narrowing of blood vessels leading to an increased blood pressure. Ongoing research is focused on reducing these effects of improving stability. So the dream of perfect artificial blood is still progress, but every experiment brings us closer to that goal. And today, this lecture, we saw about what is the artificial blood, what is the need for that, and what are the ideal characteristics for the artificial blood, and the types of artificial blood like perfluorocarbons and hemoglobin-based oxygen carriers, and then what are the manufacturing process are there, and what are the limitations are there. And these are the references. Thank you. Have a great day.